The ability of organisms to produce more of their own distinguishes living things from non-living matter, because continuity of life is based on cell division. As we move forward, be able to describe the differences between prokaryotic cell division and eukaryotic cell division. Prokaryotic cell division is a simplified version known as binary fission that involves the cell getting larger, the copying of the DNA, and then division. Eukaryotic cell division can occur one of two ways: mitosis or meiosis. As we move forward, we'll discuss each of these in turn. The goal for you is to be able to recognize the products of these two pathways and which cells undergo it. Since prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. This process is also asexual, resulting in two daughter cells with the complete genome, creating near identical clones. There's always the potential for mutation and error, but mitosis and binary fission, the goal is to create an exact duplication. Notice that all cells in your body can undergo mitosis. These are known as somatic cells, your basic everyday body cell. This is in contrast to meiosis, which involves only the specialized gametes, sperm or egg. Meiosis is specific to sexual replication, resulting in 4 daughter cells, each of these having only half the genetic content. Some terms to refresh as we enter into our conversation about cell replication and genetics. Haploid versus diploid. Haploid meaning half. This refers to the 4 daughter cells of meiosis that get only half the genetic components, one single set of chromosomes. Diploid, like a pair of dice, refers to 2 sets, the full genome. When we refer to a chromosome, it's important to be able to visualize that the chromosome is each made up of 2 chromatids, and they are held together by a centromere. Now let's talk about the cell cycle. The cell cycle consists of 2 phases, interphase and the actual mitosis. The significance of interphase is that this is where the cell will spend 90% of its life cycle, just preparing for division, doing the regular day-to-day tasks, and getting ready to divide should that need arise. Preparations for mitosis include getting larger, building the centrosome. You might recall from our chapter on the cell components that a centrosome is comprised of 2 centrioles that are essentially rings of microtubules. And from this, more microtubules will organize to form an aster or star. This aster, through polarity and charge, will replace the nuclear envelope as a mechanism to shelter the chromosomes while movement is occurring. They are essential to cell division. And so during interphase, there are 3 subphases that take part to prepare the cell for a successful division. The first phase is known as the gap phase, or G1 phase. This is a phase of major growth. The centrioles will be formed, and the cell will physically get larger to accommodate the new duplication materials that are forthcoming. This transitions us to the S phase, S for synthesis. During this time, the DNA is synthesized or copied. So too are the centrosomes. Then we enter the second gap phase. It is a secondary growth phase. During this time, all the organelles become larger. They're being replicated. So when the cell divides, all the base organelles are also split between the 2 cells. The asters will start to form around the centrosome, getting ready to replace the nucleus when division begins to occur. During this time, the cell will continue to carry out normal functions. It just also is preparing for division. Then we can enter mitosis. Mitosis is broken down into 5 subphases: prophase, prometaphase, metaphase, anaphase, and telophase. Be aware of these 5 phases, be aware of their flow, be able to recognize the unique things that are happening within each phase. Prophase is summarized as a period where the chromosomes are going to condense. Remember that to stay organized, the laws of thermodynamics describe an input of energy must be utilized. The cell cannot afford to spend energy left and right. And so when the cell is not dividing, the chromosomes are actually loose and unwound. But as preparations occur for the cell to divide and for the chromosomes to get ready to move, they will start to fold in upon themselves, condensing down into that characteristic X shape that we know. We will also start to see the mitotic spindle start to form outside the nuclear envelope. In prometaphase, which gets its name from overlapping periods of prophase and periods of metaphase, the nuclear envelope will break down, releasing and freeing the chromosomes, to which the microtubules will flood through and begin to attach to the chromosomes, and the mitotic spindle will start to shelter them and try to buffet them from any other damage along the way. Then there's metaphase. Meta meaning middle. This is where the chromosomes will align at the center of the cell. Essentially what's happening is each centrosome is pulling gently, and because they're both bound to each chromosome pair, when they're evenly pulling, the chromosomes will line up dead center. Then there's anaphase. During this time, the kinetochores will contract and start breaking microtubules down, and this will essentially appear to reel in the chromosomes, and they will be divided from their centromeres, and half the content will go one direction and the other half will go to the other side of the cell. Then comes telophase. The nuclear envelope is going to begin to reform around the chromosomes. They're going to start to decondense during this time because it took a lot of energy to hold it tight and rigid and small. And telophase often overlaps with cytokinesis, and this is the actual division of the cytoplasm. In animals, a cleavage furrow occurs, in which case it pinches from the outside going in, creating a cleavage furrow that divides the cells in two. In plants, a cell plate forms and separates them from the inside toward the out. In either case, you now have 2 daughter cells being formed. As we build upon our understanding of cell replication, it's important to spend some time discussing the regulation. The frequency of cell division will vary with the type of cell. Eyelashes live for 3 days. Skin cells have short lifespans. Some of your heart cells have been around for 7 years before they're replaced. There are over 210 cell types in the body, and their lifespan, from being born, differentiating, their functionality, depends upon what they're doing. Timing for each of these is critical. If they divide too often, this leads to cancer and unnecessary, unwanted, unregulated growth. If they don't divide often enough, it leads to death because there's a niche or a hole that's not filled, a function not being performed. During fetal development, many cells are programmed to die. They have a shelf life. And cells appear to only undergo a set number of cell divisions, roughly 40 or 50 in cell cultures. So the potential for cell division is not unlimited, hinting at a regulatory system, a fine-tuned control system regulated by internal and external controls. Within the body, within the cell, there are enzymes that pass messages and activate a pathway to prepare the cell for division. Outside of the cell, there are growth factors that encourage new cell formation. There also has to be a need. Healthy cells exhibit something known as density dependency, and this means that they will not divide if they are crowded. They are self-inhibiting. If we look at this petri dish, when 2 cells are removed, the nearby cells begin to divide, but only so much as to refill that hole that was left. Healthy cells also exhibit something known as anchorage dependency. They've got touch sensor receptors and must be able to attach to the substrate or the basal lamina, or they will not divide. Floating in a vat of liquid, cells will just sit and wait. How do you know which cell will fill this need? Nearby cells will be stimulated to prepare for division, but each one will have to go through a series of checkpoints, allowing for the most adapted to engage in the actual division. Be aware that there are 3 checkpoints and where they occur: in gaps 1, 2, and during mitosis. The G1 checkpoint is the most important. It is also known as the commitment checkpoint. At this point, if the cell passes this checkpoint, it will go all the way through and complete the cycle. If the cell does not pass this checkpoint, it will be relegated to a limbo known as gap 0, the G0 phase. What is this checkpoint looking for? Is the DNA okay? Are these chromosomes a healthy example to be duplicated? Are there enough resources, nucleotides, to make a full copy of those chromosomes? And the environment. Is there room? Is there anchored sites? Am I getting the correct signal saying this is necessary? If a cell does not pass the gap 1 phase and goes to the gap 0 phase, it will exist in a quiescent state. I mentioned limbo. Gap 0 is essentially an extended gap 1 phase. It's a phase where the cell will exist and live and grow and perform its functionality, but it will not divide and it will not prepare to divide. Why isn't the cell just destroyed? Well, the idea is to maintain a standard number of cells in the specific tissue and conserve resources. During gap 2, this checkpoint will again look at the DNA. Was everything copied correctly? Do I have the right number of chromosomes? And then look to see if there are the right proteins present for cell division, the centrioles, enough microtubules. Do we have ribosomes and organelles ready to go? Finally, the M checkpoint will again look at the DNA, but most importantly, the M checkpoint is going to look to see whether every chromatid is attached to a spindle. This checkpoint ensures that the 2 cells produced by mitosis will end up with the correct number of chromosomes. No duplications, no shortcomings. Even with all the checks and balances and regulation, things can go wrong. Cancer is a cell cycle disease. When one of your normal cells stops listening to the correct cues, ignores regulation, we say it is transformed. It has been converted to a cancerous cell. Many cancerous cells are destroyed by your immune system within the first 24 hours. Those that are not, though, can form tumors, excessive cell growth. Benign tumors get their name for being kind. These are abnormal cells that remain in the original site, easy to excise. Malignant tumors are the unkind one. Malignant tumors are the unkind tumors. These cells will invade surrounding tissues, making it much more difficult to remove. The worst are metastasized cancer cells. These are tumors that are no longer attached at their source, but now have moved to other parts of the body, seeding new tumor-like growths elsewhere. It's important to note that as commonplace as cancer seems, a whole series of changes have to occur to lead to this disorder, and that's why it typically occurs at an older age, where time allows the compounding of these various mutations and circumstances. Due to our extended lifespans in modern society, 1 in 3 people will develop cancer in their lifetime. There is no one hallmark for transforming a cancerous cell, but we have found 6 changes that are found in most, if not all, cancers. The first being self-sufficiency. It does not respond normally to the body's control mechanisms. In this case, it makes its own growth signals, not waiting to get feedback from surrounding tissues. Insensitivity to growth inhibitors. It ignores the regulatory signals to stop dividing. Evasion of apoptosis. This means when the body is trying to target a dangerous cell to commit cell suicide and die, it is able to say, nope, not doing that either. Cancer cells often display no density dependence. They will grow over and over, through, on each other, which is essentially what a tumor is, excessive cell growth. They exhibit no anchorage dependence. They don't need to bind to the surrounding floor. They will grow sideways, upside down, inside out. Where most cells will stop dividing at 40 or 50 times, cancer cells don't. They will continue to divide, heedless to the dangers of a shortened genetic information or a mutated genetic information. And another thing that they can do is they can often encourage angiogenesis, the formation of blood vessels to feed them so that they can get first dibs on incoming resources. Cancer's not new. It's been documented by ancient Egyptians. We just didn't see it historically because many people died young. There were so many infectious diseases and wars that the average lifespan was only about 35. Not enough time for cancer to really develop and take hold. Once infectious disease was controlled, due to our public health improvements, industry, capitalization, and improved medical care, cancer has become more commonplace, especially when we see the average life expectancy increase to now 85 years of age. So where did the transformations come from? How does a cell suddenly ignore regulation or make its own growth factors? It acquires mutations, small, subtle changes that maybe allow it to function differently. Mutations occur naturally, but can also be exacerbated by radiation, harsh environments, or heredity, predisposed mutations. In fact, we find that in a vast majority of cancers, the p53 gene is altered. This gene directly coincides with the ability of the cell cycle to check the G1 checkpoint. BRCA1 and BRCA2 are the breast cancer genes. If you attain one copy of that gene, you're predisposed to breast cancer. 2 copies, and you're more likely to develop breast cancer. But it's just a risk factor, does not guarantee it. And some 11% of all cancers are caused by viruses or bacterial infections that put strain on the surrounding cells, leading to mutations and altered functions. Meiosis is basically similar to mitosis, except that there will be 2 subsequent rounds of division, having the material so that instead of the 2 cells with a complete genome that we see in mitosis, we then get 4 cells with only half the information. So in prophase 1, in mitosis, we saw chromosomes condense in a mitotic spindle form. The same is happening in meiosis. But it's important to note that in both mitosis and meiosis, this is the complete genome, the homologous chromosomes of mom and dad. Prometaphase occurs as expected with the nuclear envelope breaking down and the microtubules flooding in. Metaphase 1 proceeds as expected with the chromosome pairs aligning at the center. In anaphase 1 of meiosis, much like that of mitosis, the chromosomes are separating. We're dividing out mom's and dad's full chromosomes. And then we enter into telophase, where the nuclear membrane reforms and the chromosomes begin to unravel, and the cell pinches off and splits into 2. Now 2 cells progress through mitosis 2. The chromosomes, having had just a breather, begin to condense again, and the mitotic spindle will reform. In prometaphase, the nuclear envelope will form vesicles around mitotic spindles, but this is happening in 2 cells simultaneously. In metaphase, the chromosomes align at the center. They are no longer pairs. And so what's happening is the microtubules are attaching to each of the sister chromatids. Recombination is often called crossover because it involves 2 homologous chromosomes side by side crossing over their chromatids. And these non-sister chromatids sometimes become tangled and require repair mechanisms to remove their knots. Sometimes when they are cut and resewn together, they've exchanged DNA segments. The wrong sections have been paired to the wrong parental chromosomes. That in itself is not a problem because they're carrying the same types of instructions, eye color for eye color, hair color for hair color. The difference is that now there's some shuffling of combinations. Crossover happens between prophase and metaphase when all the chromatids and homologous chromosomes line up near the metaphase plate. Chromosomes, genes that are close together are likely to be inherited together despite crossover and any breaks that occur. So now when anaphase 2 occurs, the sister chromatids are what separate.